Hi everyone, welcome to the Genomic Technology SIG webinar number 2 in our fall webinar series. The topic for today's webinar is Advances in Single Gene NIPT. And we're very excited to have many members of other SIGs, including the prenatal SIG, joining us today. So thank you for everyone who attended or registered to attend. Just a couple housekeeping items to start off with. This is a CEU eligible webinar. So there's up to 1 contact hour or 0.1 CEUs allowed. For those of you who would like to collect a CEU for this attendance, there are a couple of requirements. One is that at the end of this webinar, there will be a survey. The survey will include both the assessment questions as well as the evaluation. Please do complete that at the end if you're able to. For those of you who are not able to complete it at the end of the webinar, a link to the evaluation and assessment will also be included in an email that will come out shortly after the webinar ends. Please be sure to complete both of those by December 6th if you would like CEU credit for this webinar in this calendar year. Also, for those of you who are not attending the full series or are only attending this webinar, there is a $25 CEU fee, which has to be paid directly to NSGC for CEUs. All right, without further ado, I'd like to introduce our speakers and encourage everyone to enter questions for them as we go through the lecture today. So within the GoToWebinar panel, you should see an option for questions, and you can enter questions there. And at the end, if we have time, which I'm sure we will go over those questions with our speakers. So let me introduce who we have with us today. Sorry about my dogs. So Brian Landry, or Dr. Landry, is the principal scientist at Billion to One. He was trained at Vice University and has his PhD in bioengineering, and he was actually the first employee at Billion to One, and so he's been really heavily involved in the NIPT space. We also have presenting today Sarah Reardon. Sarah Reardon is a genetic counselor. She has been practicing for over 15 years. And she consults across multiple disciplines, including oncology and prenatal, and she is a consultant for Billion to One. So welcome, both of you, and I will turn it over to Sarah.
Great, thank you, Shannon. And thank you to the Genomic Technology SIG for having us speak today. I love the fact that the SIG is really dedicated to understanding new genomic technologies and learning about those technologies. And certainly we are really excited to talk about the technology that Billion to One has developed, which is a molecular counting technology, and also excited to talk about the test, our first test that technology enables, which is a carrier screen with reflex single gene NIPT. And just our disclosures, I'm Sarah Reardon, I'm a paid consultant of Billion to One. I will be passing off the buck to Brian Landry at some point in the presentation, and he's going to talk a little bit more about our technology, and Brian is an employee of Billion to One. So we'd like to just start off by addressing sort of the current state of the state with prenatal carrier screening and some of the unmet needs that we're trying to solve with the test. So ACOG recommends offering screening to every pregnancy for sickle cell disease, cystic fibrosis, beta thalassemia, alpha thalassemia, and more recently they added SMA to this list in 2017. And these conditions really sort of fit into kind of these hallmarks of ideas that we want to address with carrier screening. First is frequency. So the carrier rates for these conditions, when we add them up together, about 11% of people, and of course this varies across ethnicities, but on average about 11% of people are carriers for these conditions, so they are common. To a large degree penetrant. If you have a variant, express some degree of the disease that actually affects quality of life significantly. And they have early onset, very early in childhood. And there's actionability with these conditions in that there are treatments that can either cure the condition or ameliorate symptoms. And so all of these conditions sort of fit these hallmarks, which is why CONCIT recommends that every woman be screened for these conditions. But yet, we're really seeing some large unmet needs with carrier screening. So a review of looked at the adoption found that only 46 to 78% of pregnant women in the United States will actually accept CF carrier screening when it's offered by their provider. With SMA, it's much lower. This was a study that looked at over 1,000 women who were offered SMA screening during their pregnancy by their provider, and the uptake rate was 20%. That was actually before ACOG recommended that every woman be screened with SMA, but still we're seeing really low adoption rates of this. And despite the fact that there for SMA, and most the newest was Zolgensma was just released this, or just approved by the FDA this year. Only 5 states are offering newborn screening for SMA. Another 13 are piloting this. So we are not, we're not seeing adoption during pregnancy of carrier screening, and then we're not actually capturing a lot of these babies. And these treatments are really most effective when they're started as soon as possible after birth. So definitely some unmet needs. And when we look at the sort of carrier screening workflow, so typically we're going to offer mom carrier testing early in pregnancy, one of her first prenatal visits, and 11% are going to test positive, are going to have a positive carrier screen. But most of these women will end up having healthy babies, so we're causing some worry and anxiety there until dad can get in for screening. And we know that this step just doesn't always happen. And there's not great data in the published data in this area, but data from various clinics where it looks like compliance with maternal carrier screening is less than 50%. I saw a couple of posters at the NSGC AEC that also reflected this. And the reality is that 40% of moms in the US are single mothers. We have non-paternity issues. Just too busy to get in for their screening. And so we don't always complete this process. And when dads do come in for screening, this adds extra time. And so often we're getting up to 20 weeks of pregnancy before we actually have both parents' carrier screening results and then offering invasive diagnostic testing at that time. What the test that BillionToOne has developed is trying to address is to actually eliminate some of these steps. And so this is a carrier screen that automatically reflexes to single gene NIPT if mom is a positive carrier. So it does not require paternal DNA at all. It's 1 tube of maternal blood as early as 10 weeks of pregnancy. And because only about 1% a positive carrier screen and then a high-risk NIPT result, we're really offering invasive testing to only the most high-risk patients. And so this is a process that we can complete much faster. We're reducing a lot of anxiety and worry, and we are reducing unnecessary clinic follow-up. So looking just in more detail at the carrier screen with reflex single gene NIPT, this is a test as carrier screen for cystic fibrosis, spinal muscular atrophy, sickle cell disease, alpha thalassemia, and beta thalassemia. When mom is positive, a positive carrier, it reflexes to single gene NIPT for those conditions. As part of the test, we also offer a 43 gene expanded carrier screen panel for patients of Ashkenazi Jewish, French Canadian, and Cajun descent. We don't actually reflex to NIPT for those additional genes, but the additional carrier screening is available. It takes 1 tube of blood starting at 10 weeks of pregnancy, performed on singleton pregnancies only. And the technology is NGS based. We do MLPA for deletion analysis. And the carrier screening rates are pretty typical or better than what we see across the industry. We're looking at all exons, exon-intron junctions, select introns for CFTR and HBB. For alpha thalassemia, we do report the silent carrier. And for SMA, we do report the SNP that tags the silent risk for silent carrier. And Brian is going to talk a little bit more about our analytical validation and clinical trials, but the NIPT has demonstrated greater than 98.5% analytical sensitivity and greater than 99% specificity. And we've seen 100% concordance with newborn screening in our 2 clinical trials. So I've kind of thrown around the term single gene NIPT like it's no thing, but this is actually a new technology. So when we think of NIPT in general, we usually think of it with and so looking at trisomies or sex chromosome aneuploidy. And the way this is typically done is we measure highly conserved regions along a chromosome, and we count up those regions. It's quantitative, but we're looking at many sites across a chromosome. Single gene NIPT is looking at one single site. And we have seen this done with dominant conditions. There are a few labs offering single gene NIPT for de novo mutation, just for the presence or the absence of a mutation. So it's qualitative.
Sarah, I'm sorry to interrupt.
With single gene NIPT.
Sarah, are you able to hear me? You're cutting in and out quite a bit for us.
Oh no, okay.
Sorry, just a few commented that we're having trouble hearing you. So I just wanted to see if there's any better connection spot maybe for you.
Yeah, let me try to move locations.
Thanks, sorry to interrupt.
Give me just a second. Okay, can you hear me okay?
Yes, can hear you now.
Okay. All right, so let me just back up a little bit. So just sort of looking at how NIPT is traditionally done. Aneuploidy NIPT is what we're sort of used to seeing, right? This is measuring many locations across a chromosome. Single gene NIPT for dominant conditions is really just, it's qualitative. It's measuring the presence or absence of a de novo mutation. The challenge with single gene NIPT for recessive conditions is that it's a single site, and it also has to be quantitative, because we know that mom is a carrier, right? And she has her carrier variant, and we're trying to understand how much of a variant from the fetus is present against mom's background. And so the challenge with single gene NIPT for recessive conditions is actually getting that fine-tuned quantitative data at a single site. And Brian is going to talk about the proprietary technology that Billion-to-One has developed that enables that. So looking just at sort of at the technical workflow for how the test works, we start with maternal blood, at least 10 weeks gestation. And from the plasma, we isolate genomic DNA. We do MLPA or NGS for the carrier screen. If we find a variant, we confirm that by Sanger. So pretty standard there. If we identify mom as a carrier, then we reflex to the single gene NIPT. So from the cell-free DNA, we do actually several different NIPT assays. And so I'm going to kind of walk you through the different assays that are done depending on the condition. So the relative mutation dosage is what I was referring to on the last slide, where we actually have to dose how much of a variant, dose the presence of a fetal variant against the maternal background. But then we also want to understand if there is a paternally derived variant in the fetal fraction. And so we do both of these assays, the relative mutation dosage and the detection of the paternal variant, for CFTR and HBB for sickle cell and beta thalassemia. And the relative mutation dosage can tell us if the fetus has inherited mom's mutation, and if fetus is actually homozygous for the same if the fetus inherited the same mutation from both mom and dad, let's say Delta 508. But we also run this other assay in order to determine if the fetus detected a different variant from dad. So if fetus is a compound heterozygote. For SMA, we do copy number analysis. And for alpha thalassemia, we do deletion analysis to detect paternal inheritance of the HBA1, HBA2 deletion in cis. We also, for alpha thalassemia, perform relative mutation dosage to detect inheritance of the constant spring allele. So just to review a little bit more detail, what we detect with the NIPT. For alpha thalassemia, we're looking at for common double gene deletions. We also look for presence of constant spring. The NIPT for alpha thalassemia is optimized to detect the most severe forms of alpha thalassemia. So HB-BART, constant spring, we're not see if baby is at risk for non-constant spring HBH disease. For beta thal and sickle cell and cystic fibrosis, we do NGS and are able to detect most variants, greater than 90%. And for SMA, we do copy number analysis. While the carrier screen will pick up the SNP that tags the silent carrier, we actually don't reflex to NIPT for that. It's really just the math. SNP actually detects is probability. So in order for the math to work out, we have to actually know if mom is a carrier. So would like to walk through a couple of sample reports before I pass it over to Brian, who's actually going to review some specific cases. So when we don't detect that mom is a carrier, the negative carrier report looks very similar to what you're used to seeing with carrier screening. These reports are when we detected that mom is a carrier. Now on the left, we detected mom as a carrier, but low risk fetus. So the baby has not inherited a paternally derived cystic fibrosis variant. And so we're able to report a very low posterior risk, less than 1 in 9,600. On the right-hand side, we have a report when we've identified mom as a carrier, we've reflexed to NIPT and identified baby as high risk. So here we've identified that the baby is likely homozygous for inherited the same variant from mom and from dad. And so the posterior risk is greater than 9 in 10 that this child is affected with cystic fibrosis. And that posterior risk is a personalized risk, and it can vary a little bit depending, the range of posterior risk can vary a little bit depending on the condition and the specific mutations involved and several other factors. When we look at sort of a breakdown of our high risk results, the vast majority are going to be greater than 9 in 10, greater than 90% risk. Less than 10% of the time we have, will present numbers that are somewhere in between 1 in 2 and 9 in 10. And again, that varies on several factors, including the condition and the specific variants that are involved. Our recommendation for all high risk results is the same, which is to offer invasive diagnostic testing and genetic counseling. Billion to One does have genetic counseling on staff that can provide pre and post-test counseling at no charge. And so at this point, I'd like to turn it over to Brian, who's going to go over some really interesting cases, talk a little bit more in detail about the technology, and also talk about some of our clinical trial results. So let me pass it over to you, Brian.
Hello, everyone. It's good talking with you today. I'm the principal scientist at Billion to One, and these single gene NIPTs that we offer, I was the lead scientist in developing them all. So I'm really excited to kind of share the work that I've done with all of you today. I want to actually start off by circling back to a slide Sarah was just presenting, describing the different types of non-invasive prenatal tests that exist. So how these non-invasive prenatal tests work is that cell-free DNA is purified from a pregnant mother's blood. This cell-free DNA actually contains a mixture of the mother's DNA as well as the fetus's DNA. Currently, this is widely used to detect trisomies. This is actually a fairly easy problem technically because chromosomes are millions of base pairs long, and so it's very easy to detect if one of them is elevated compared to what is normal. The problem that we've faced at Billion to One in developing these single gene NIPTs for recessive conditions is that we have to look for a single base mutation in one of these genes to determine whether or not the fetus has inherited it. And not only are we looking at a single base, but we have to determine if that base is coming from the fetus or from the mother, because the mother is a heterozygote carrier for that mutation. What this means is that we've had to develop new technologies that allow us to perform very accurate molecular counting on how many molecules of a healthy allele or a mutated allele are present in a cell-free DNA sample that we receive from the patient. So the way non-invasive prenatal tests work is the DNA is purified, the cell-free DNA is purified, and then next generation sequencing is performed upon it. However, before you can perform next generation sequencing, you actually have to amplify the cell-free DNA over 1 million-fold. And this creates an issue where we actually don't know exactly what this amplification factor is. And without knowing this amplification factor, we can't calculate how many mutated DNA molecules and how many healthy DNA molecules we started with in the cell-free DNA that we purified from the patient sample. And so the challenge we faced in developing an extremely accurate single gene NIPT was being able to measure this amplification factor for every single patient sample. And therefore, once we know the amplification factor, we can actually calculate how many mutated DNA molecules we started with, and therefore make a conclusion on the genotype of the fetus. And so what I've helped develop is a new technology called quantitative counting templates. Quantitative counting templates are artificially designed DNA molecules that amplify at the same rate as the genes that we are looking at. However, these quantitative counting templates are designed such that every single molecule has a completely unique DNA sequence. What this means is that we can only ever add one quantitative counting template to each cell-free DNA sample when we prepare it and perform next-generation sequencing. Therefore, we know we've added exactly one quantitative counting template to every sample. So when we get the sequencing output from that quantitative counting template, that actually equals our amplification factor. Therefore, this enables us to measure an amplification factor for every single patient sample using quantitative counting templates. And so we don't even just do this once for a patient sample. We do this over 300 times. And so we'll add in 300 of these different quantitative counting templates. And to repeat, these are DNA molecules that we've artificially designed that allow us to calculate this amplification factor for the gene that we are looking at. And so we make 300 different measurements of this amplification factor for every single patient sample. Once we've measured this amplification factor for a patient sample, we can then look at the sequencing data for the mutated gene in the cell-free DNA. And using the sequencing output, as well as this calculated amplification factor, we can determine the exact number of mutated DNA molecules and wild-type DNA molecules that we started with. To give you a sense of how this fits into our overall workflow, this is kind of an overview of how our single gene NIPT assays function. So after we've identified that a mother is a carrier, we go back to that same blood tube, take out the plasma from that blood tube, and purify the cell-free DNA. So this cell-free DNA contains a mixture of both the mother's and the fetus's DNA molecules. We then add these quantitative counting templates, which are these artificial DNA molecules that amplify at the same rate as the cell-free DNA that we're looking at. With this mixed sample, we perform amplification and next-generation DNA sequencing on the exons of these genes. Then we can look at the quantitative counting templates and the sequencing data to determine what the amplification factor was for that patient sample. Once we've calculated the amplification factor, we can then go look at the sequencing data and determine exactly how many molecules of mutated or wild-type DNA we started with in that cell-free DNA sample. We can then use that molecular count information to infer the probable genotype of the fetus. This is a really exciting new technology that I was really proud of being on the team that created it, and is enabling us to perform all of these different single gene NIPTs that we do. We recently published an analytical validation of this technology in Nature Scientific Reports, and we showed that we had 98.5% analytical specificity and 99% analytical sensitivity. So now that I've kind of explained this technology, I want to walk through a couple case studies that show it in action and kind of explain to you how using this molecular count information, we're able to infer the genotype of the fetus. So this is data from an actual clinical sample that we received from a hospital. So when we received the sample, we performed our standard carrier screening on it. This carrier screening identified that the mother was a carrier for cystic fibrosis. The mother was a heterozygote for the F5 or 8 deletion in the CFTR gene. Because this mother was a carrier for cystic fibrosis, we automatically, using that same tube of blood, reflex to the CFTR single gene NIPT. What this means is we purified this mother's cell-free DNA. We added in the quantitative counting templates. We performed next-generation sequencing on the exons of CFTR. And then using the quantitative counting template sequencing information, we calculated an amplification factor for this patient sample. Using this amplification factor, we're able to determine exactly how many molecules of the normal CFTR gene and how many molecules of the F508 deletion CFTR gene were present in the cell-free DNA. And so you can see on the right that we determined that there were 1,457 molecules of the normal CFTR gene, and that there were 1,364 molecules of the F508 deletion CFTR gene. So looking at this data, we can see that the mother is heterozygote, obviously, as the carrier screen had shown, but that the fetus was also likely a heterozygote for the F508 deletion. This tells us that the fetus is not an affected F508 deletion homozygote. We also performed next generation sequencing on other exons in the CFTR gene and found that the fetus was negative for a compound heterozygote affected state as well. To give confidence to these results, we also measured the fetal fraction, as we do with every sample, and found that this had a 15% fetal fraction. What this allows us to do is use our statistical models to determine that the fetus has an extremely low risk of having cystic fibrosis, despite the fact that the mother is a carrier. And so you can see this is the report that we've returned to the mother. This report says that the mother is a carrier for cystic fibrosis, that she has this F508 deletion. However, in the exact same report, we can say that the fetus has a low risk for having this disease, less than 1 in 2,000 chance. So you just kind of very broadly, you can see how this is a much better experience than traditional carrier screening. If this was a traditional carrier screening result, the patient would have just learned that she was a positive carrier, and therefore her child had a significant risk of having this disease. She would have then had to hopefully pursue follow-up testing of the father or more invasive testing to determine whether or not her fetus actually was at a high risk for having cystic fibrosis. This alternative report that we are providing says at the same time that the mother is a carrier for cystic fibrosis, but the fetus is unlikely to have inherited the disease. We followed up with this patient, and using newborn screening results from the child, we confirmed that this child was negative for cystic fibrosis, as our test predicted. In fact, we were able to get the genotype of the child, and the child was a heterozygote for that F508 deletion, just as the data in the previous slide suggested. So this is kind of the first of 3 case studies I want to present to you today. The next 2 are actually from our clinical trials, which I want to spend a little time talking about now. So before we launched our test commercially, we performed a clinical trial with Yashoda Hospital Specialty Clinics. As a part of this trial, we performed 27 NIPTs on pregnant mothers. We then collected follow-up newborn screening data for every single one of those pregnancies. We compared the newborn screening data with the non-invasive prenatal test result that we had generated, and found that every single one of those test results were correct. This data has recently been published in Nature Scientific Reports if you'd like to take a look at it. We've actually followed this first clinical trial up with a second ongoing clinical trial. So we applied for and received funding from the National Institute of Health to conduct a larger scale clinical trial. We are pursuing this with Baylor College of Medicine, as well as the University of Alabama. And by 2020, we hope to have over 100 samples in this clinical trial. We've performed interim analysis of the first 50 samples that we received as a part of this clinical trial, and found that 100% of our NIPT results were concordant with the newborn screening result for those children. Interestingly, in this clinical trial, we actually predicted both a positive case for sickle cell disease in a fetus, as well as a positive case for the compound heterozygote HbS-C sickle cell disease. Both of these positive non-invasive prenatal test results were confirmed with newborn screening. Here I want to present a second case study that came from this clinical trial. In this situation, when we screened the patient for all of the ACOG recommended disorders, we found that she was a carrier for sickle cell disease. What this means is that this patient has the HBS allele in the HBB gene that can cause sickle cell disease. Once we identified that, we automatically reflex to a single gene NIPT of the cell-free DNA from the same blood tube we performed the carrier test on. Once we've purified out the cell-free DNA, we added in those quantitative counting templates, which are the artificial molecules that amplify at the same rate as the cell-free DNA. Using the quantitative counting template technology, we're able to calculate an amplification factor for this patient sample. We then use that amplification factor to determine how many molecules we started with of the normal HBB sequence and how many molecules of the mutated HBS sickle cell disease HBB sequence we had in that cell-free DNA. Looking at the data, we found that there were 1,422 molecules of the normal HBB gene, and that there were 1,423 molecules of the mutated HBS HBB gene. Based on this data, we are able to predict that the child was likely a heterozygote for the HBS allele and therefore was not affected with sickle cell disease. We also screened other exons of HBB to determine that the child was also likely negative for being a beta thalassemia compound heterozygote. We also measured the 9.3% fetal fraction that this sample had to determine that we could return these results with high confidence. Based on these data, we are able to return this report to the patient. This report says that the mother is a positive carrier for sickle cell disease. However, at the same time, it says that the fetus has a low risk for having this disease. And again, you can see how this is just a much smoother workflow than the traditional carrier screening process. We followed up with this newborn, and we confirmed that our call was correct and that the newborn was negative for sickle cell disease. Now, what's interesting in this case, as I showed on the previous slide, is that we were able to predict that the child was likely a heterozygote for the HBS allele. And there's actually a slight phenotype associated with being a heterozygote for the sickle cell allele. That phenotype is called sickle cell trait. And so based on our data, we could internally say, hey, this fetus is likely to have the sickle cell trait phenotype. And the follow-up screening actually confirmed that the child was heterozygote and therefore affected with sickle cell trait. Now, sickle cell trait is not something we report because it is not nearly high enough severity to be important to report as part of a carrier screening process. However, it is something that we can predict with our single gene NIPT. And what's nice about predicting the sickle cell trait status as a part of this clinical trial is it kind of lets us do another test of the capability of our single gene NIPTs. And so for all of the HBS carriers in this clinical trial, we are reporting a predicted sickle cell trait status of the fetus. And so what we're able to do here is report both negative sickle cell trait status. And so this is when the child has completely wild type HBB alleles, or positive for sickle cell trait. And this is when the child will be heterozygotes for the HBS allele. And we do this because we expect a large number of both negative and positive results for sickle cell trait as a part of this trial. And in fact, this is what we've seen. Our single gene NIPT has predicted over 20 negative results for sickle cell trait and has also predicted over 20 positive results for sickle cell trait. Every single one of those results from our single gene NIPT has been shown to be concordant with the newborn genotype. This has been fantastic data for me to see that these single gene NIPTs that we have developed are extremely accurate at predicting both negative and positive results for these diseases that we look at. I want to end the portion of my talk with one of the more interesting cases that we've looked at as a company. And so this was another sample that we received as a part of this clinical trial. When we performed carrier screening on this sample, we found that the mother was actually likely affected with sickle cell beta thalassemia. What this means is the mother was a compound heterozygote. So one of her alleles was the standard HBS sickle cell allele. However, her other chromosome also had a large HBB deletion. So the entire HBB gene was deleted on her second chromosome. This means that the mother's not just a carrier, but she's also likely affected by this disease. Our single gene NIPTs are actually very capable of dealing with complex situations such as this one. And so for this sample, just like any other sample, we automatically reflexed to a single gene HBB NIPT. We purified the cell-free DNA from this mother's blood, and then we added in our quantitative counting templates. We used the quantitative counting templates to measure the amplification factor for this patient sample. And then using that measured amplification factor, as well as the sequencing outputs for the cell-free DNA HBB gene, we calculated exactly how many molecules we started with in the cell-free DNA for the different variants of this HBB gene. What this showed us is when we looked at the sequencing data, we found that there are actually 491 molecules of the normal HBB gene, and that there are also 3,469 molecules of the mutated HBS HBB gene. Now, if you remember, this mother is a compound heterozygote for HBS and an entire HBB gene deletion. So what that tells us is all 491 molecules in that fetal DNA that are the normal HBB gene had to come from the father and therefore were inherited by the fetus. And so looking at this data, we can very conclusively say that the child inherited a normal HBB molecule and therefore will not be affected with sickle cell or beta thalassemia. We also measured the fetal fraction for the sample at 13.2% in order to give us confidence that we can return this result and that these DNA molecules we're seeing are coming from the fetus as well as the mother. We are then able to return this report to the patient, and that is saying that this patient is a positive carrier. And in fact, in the details in here, it says the patient is likely affected with sickle cell disease. Even though this patient is likely affected with sickle cell disease, we are still able to say, hey, this is a very low risk pregnancy for the fetus. In this case, it was a 1 in 2,000 risk for the fetus to have sickle cell disease. As with the other examples, we followed up this result with newborn screening and confirmed that the fetus was negative for sickle cell disease. This is the end of the case studies that I'm presenting to you, but I really kind of want to emphasize how much of a more streamlined carrier screening experience this is by having the patient receive both a positive carrier result, but also a low risk fetal result for many of these pregnancies. Something that I'm really happy to have been a part of and really kind of proud of creating this technology that's enabling these new single gene NIPTs. So now I want to pass it back to Sarah, who is going to present a few summary slides to kind of go over this technology and how it integrates into the carrier screening workflow.
Great, thank you, Brian. Okay, so I want to address the question that I think many of you are wondering, and we've certainly gotten this inquiry from lots of genetic counselors, is, okay, you're doing single gene NIPT for recessive conditions. Is this a non-invasive option for my known carrier couples? And of course, generally, when you have a 25% risk to have an affected child, we recommend invasive diagnostic testing. But of course, we have lots of patients who don't want invasive testing. And so, is this an option? And our answer is, hold on, sorry, my screen is not advancing. There we go. Our answer is yes, with some considerations. So this test is really designed to be a screen for the general population. It's not a diagnostic test. And the algorithms really best support starting out with a very low a priori risk, so kind of a general population risk. And I mentioned that the posterior risk can vary depending on the condition and the specific variant. So generally, most of our high risk results are going to be in the 9 out of 10, 90% range. But there are times when we can see lower numbers than that. And so if you're starting out with a 1 in 4 risk, getting a high risk result that is 1 in 2 is maybe not all that informative. Of course, if you go from a 1 in 4 to a 9 in 10 risk, that can be informative. If the NIPT reveals a low risk for the fetus of less than 1 in 10,000, then that's very informative. So it really just depends on the condition and the specific variants that are involved. And so we urge you just to contact us with your particular case. We can actually review what variants mom and dad have, and then give you a range of numbers that we would provide on the posterior risk side, so that you can go back with your patient and have the discussion in terms of whether this is going to be an informative test. And then, of course, there's always gene surance issues. We bill this test as a carrier screen. And so known carrier couples who have they've already had carrier testing, if insurance isn't going to cover it, then we do have an affordable cash pay price. You're welcome to contact me and I can discuss what that price is. And so just to summarize what this carrier screen with reflex single gene NIPT looks like in clinical practice, and to summarize some of the points we've gone over today, this is a way to identify the truly high-risk pregnancies early and accurately. This tests mom and baby at the same time for recessive conditions recommended for screening by ACOG, high sensitivity and specificity from the NIPT. So we're offering invasive diagnostic testing to really only the highest risk pregnancies, and we're enabling early management for pregnancies at risk for CF, SMA, and hemoglobinopathies. This really eliminates barriers that are seen in traditional carrier screening. We don't need paternal DNA, less than 2-week turnaround time for both the carrier screen and the NIPT, and we're really reducing unnecessary clinic follow-up. And as I mentioned, we bill using standard carrier screening codes specifically for cystic fibrosis and SMA. For patients who don't want to go through insurance or don't have insurance, we do have a financial assistance program, and it's a discounted self-pay option. And I mentioned that we do have complementary genetic counseling available for all patients. So that wraps up our planned slides. Again, thank you to the GT SIG for having us, and I'd like to open it up for any questions at this time.
Thank you, Sarah, and thank you, Brian. That was a great talk. Very interesting technology. We do have a couple questions from the audience. So I'm just going to start from the beginning of our questions, and I think this one was going back to Brian's slides. The question is, the 100% concordance means that you followed up to check for false negatives. So are you actually checking only the positives or also follow up on negatives?
Yes, we are following up on all of the cases. So we are looking at, for every single NIPT we perform as a part of that clinical trial, we are following up and getting the newborn screening data. So this is regardless of whether or not that NIPT was a negative result or a positive result.
Great. And then I think this was also for you, Ryan, and apologies if you mentioned it, but what is the acceptable threshold for fetal fraction?
So currently our acceptable threshold for fetal fraction for those NIPTs is 5% fetal fraction when we have to quantify the number of DNA molecules to determine recessive inheritance. For certain carrier genotypes, we actually don't necessarily have to determine recessive inheritance. And so for those situations, we can actually lower the fetal fraction cutoff to 2%. If a sample doesn't meet our fetal fraction cutoff, what we'll do is we'll return the carrier result and then at the same time ask for a redraw in hopes that the redraw will provide a high enough fetal fraction such that we can then return NIPT result.
Great. And a couple more questions, and I'm not sure who might be best to answer this one. Is there anything more that you can say about the cash pay pricing, or is it an individual case-by-case basis?
It's not individual case by case. We have an established cash pay price. It's just not something that we publicize, but please contact me and I can go over that with you.
Okay. A couple folks have asked if there are plans to include other genes, or to expand the panel offering.
That is something we are very actively considering, and actually I'd be very interested to hear what people's thoughts are on what specific genes they would be interested in, because that's something our technology is very capable of doing. It would just take time to do the research and development needed to create new single gene NIPTs for other disorders. But that's definitely something that is in the pipeline at Billion to One of thinking about what other diseases are important to target. And I will say we very much try to kind of follow the recommendations of ACOG and ACMG. And so we're very much looking at what they recommend testing for carrier screening in order to guide our thought process of where it's important to focus our single gene NIPT development resources.
Great. And can you just review again what is quantitative counting templates? What are they and why are they important in this process specifically for Billion to One?
Yeah, I'd love to. So that's really kind of the core of our technology. So these quantitative counting templates, they're DNA molecules that we synthesize. These DNA molecules are very similar to the genes that we're looking at. So a quantitative counting template may look very similar to the first exon of the HBB gene where the HBS allele is found. However, when we synthesize these artificial DNA molecules, we synthesize them in such a way that there will be a sequence of unique DNA bases in that DNA molecule. Those unique DNA bases make that molecule entirely unique. There's only one of that molecule that ever really has existed. And so what that tells us is when we add quantitative counting templates to the cell-free DNA, we're adding 300 different unique molecules, all of which start out with a copy number of 1 in that sample, so a molecule count of 1. Since these molecules are made to be very similar to, let's say, the HBV gene, when we perform amplification of the cell-free DNA and these quantitative counting templates in the same reaction, they all amplify at the exact same rate. Since they all amplify at the exact same rate, we can use the data we get from the quantitative counting template DNA sequencing to determine what this amplification factor is for that patient's cell-free DNA. And this is because these quantitative counting templates are so similar to the gene that we are sequencing. And so using these quantitative counting templates to really measure that amplification factor allows us to use fairly basic algebra to back calculate how many molecules of the wild type or the mutated gene we started with in that same cell-free DNA sample that we added the quantitative counting templates to. And so very broadly, they're just a way to get very accurate molecular count information of every single patient's cell-free DNA sample.
Great. Thank you, Brian. We have time for just a few more questions, and they keep coming in. So let me just jump ahead here. So have there been any cases in the trial so far that detected an abnormal paternal allele in the fetus? And if so, how would they report that if the fetus inherits the paternal and not the maternal allele?
Yes, so we have definitely seen paternal alleles in our sequencing data. And so the way that it is reported to the patient is if we can determine that the fetus did not inherit the mother's allele, but we did see a paternal allele come up that's different from the mother's allele, we can still return a low-risk fetal result. However, we'll also note that a paternal variant was identified in the NIPT when we performed it.
Okay. Someone is wondering if this technology could work for trinucleotide repeat disorders.
That will be difficult, but that's definitely a problem that we have been thinking about as an R&D team here. And this technology could be applied there. However, there's just a lot of complications with the trinucleotide repeats and assaying for them. So it's something that would also require kind of some new development as well. But it is something that we are kind of very actively thinking about here and considering building products around.
Okay. And Sarah, maybe you can address this one. How do you anticipate the uptake for this form of carrier screening will compare to the current hundreds of gene panels that are being used today? Do you think that patients and providers will appreciate the differences and the benefits?
Such a good question. So, right, this is not, the mother of all carrier tests. This is for every condition under the sun. What we really think that this test is ideal for is a general population carrier screen where we're really just trying to capture the conditions that are most relevant, that are most common, that affect the most amount of people. And so
Nope, we might have lost her. All right. Well, I think we got the general answer there. So that concludes our questions segment. I want to just remind everyone on the line that there is a handout available here within the webinar before you leave. The handout covers the objectives of today's webinar so that you can officially evaluate how well we met them. Also, as soon as we end this webinar, you'll be prompted in the same screen, don't close your browser, to take the assessment and the evaluation right now if you want to get it over with and out of the way. Otherwise, you will receive an email with a link to complete those. And please do get all of your paperwork done by December 6th if you would like CEU credit in this year. And you'll be receiving outreach from us as well in the NSGC about the $25 fee for the CEUs. And with that, I want to say thanks to everyone who joined us today. This was a really well-attended event. And thank you to Sarah and thank you to Brian for your time.
Thank you, everyone.
thank you so much, everyone.
Have a good day. Happy Monday.